Hi, my name is Jacopo Boni and I'm going to present you our paper just published in EMBO Journal entitled Concurrence of FGFR1 mutations modulates oncogenesis in glioneuronal tumors. Fibroblast growth factor receptor 1 is a protein belonging to the family of receptor tyrosine kinases. When activating through binding with ligands followed by autophosphorylation, these receptors can switch on and off multiple cellular responses, regulating how cells divide and differentiate during tissue development. Because of this central role, genetic alteration in FGFR1 have been linked to human diseases, from congenital disorders to cancer. In particular, activating hotspot mutations have been frequently identified in different types of brain lesions, especially in glioneuronal tumors, which are developmental forms of brain tumors affecting children and young adults. Despite the low mutation burden of these tumors, secondary mutations have been surprisingly identified together with one of the two hotspots in an increasing number of cases. In this study, we set out to understand how these different FGFR1 mutations, both single and combined, affect the function of the receptor and contribute to tumor development. To do this, we use advanced molecular and cellular approaches, including bio-ID profiling, which allows to map the network of FGFR1 proximal interactors, and glial cell models genomically modified through CRISPR-Cas9. We found that the two hotspot mutations make FGFR1 overly active and unusually stable inside cells, leading to hyperactivated intracellular signaling. In fact, oncogenic mutations help the receptor to avoid normal degradation processes, leading to protein accumulation and continuous activation of growth-promoting pathways, driving abnormal proliferation. Strikingly, the presence of secondary mutations in combination with one of the two hotspots partially restores normal receptor turnover and reduced excessive accumulation of oncogenic signaling. Thanks to our genetically modified cell models, we found that one of these co-occurring mutations, which we previously discovered in families with hereditary forms of glioneuronal tumors, did not activate growth signals but instead caused strong defects in cell self-renewal and altered the expression of key developmental genes involved in brain and glial cell differentiation. This time, the presence of hotspot mutation rescued the proliferative capacity in double mutant glioma cells. These balancing effects suggest that the combination of mutations fine-tunes FGFR1 activity to a level that is tolerated during brain development but still sufficient to promote cell growth later on and therefore oncogenesis. Altogether, our findings reveal a previously unrecognized mechanism in which co-occurring mutations in FGFR1 gene cooperate to shape tumor behavior.